This video tutorial is about the process and site of glycolysis. Glycolysis is a biochemical pathway that occurs in the cytoplasm of all living organisms that respire, including many prokaryotes. The pathway involves a sequence of 10 reactions, each catalyzed by a different enzyme, some with the help of the coenzyme NAD. You only need to know the pathway in outline, so we will consider the three main stages. Firstly, phosphorylation of glucose to hexose biphosphate. Secondly, the splitting of each hexose biphosphate molecule into two triose phosphate molecules. And thirdly, the oxidation of triose phosphate to pyruvate. The image on the slide in front of you summarizes the process of glycolysis. We're now just going to look at the structure of NAD. Enzymes that catalyze the oxidation and reduction reactions need the help of coenzymes that accept the hydrogen atoms removed during oxidation. NAD is a non-protein molecule that helps dehydrogenase enzymes to carry out oxidation reactions. NAD oxidizes substrate molecules during glycolysis, the link reaction, and the Krebs cycle. NAD is synthesized in living cells from nicotinamide, or vitamin B3. The 5-carbon sugar ribose, the nucleotide base adenine, and 2 phosphoryl groups. The image on the slide in front of you represents its molecular structure. The nicotinamide ring can accept 2 hydrogen atoms, becoming reduced NAD, or NAD. Reduced NAD carries the protons and electrons to the cristae of mitochondria and delivers them to be used in oxidative phosphorylation for the generation of ATP from ADP and P. When reduced NAD gives up the protons and electrons that it accepted during one of the first 3 stages of respiration, it becomes oxidized and can be reused to oxidize more substrate in the process, becoming reduced again. We're now going to look at the 3 main stages of glycolysis, starting with phosphorylation. Glucose is a hexose sugar, which means it contains 6 carbon atoms. Its molecules are stable and need to be activated before they can split into 3 carbon compounds. Firstly, one molecule of ATP is hydrolyzed and the released phosphyl group is added to glucose to make hexose monophosphate. Secondly, another molecule of ATP is hydrolyzed and the phosphoryl group added to the hexose phosphate to form a molecule of hexose biphosphate. This sugar has one phosphate group at carbon atom number 1 and another at carbon atom number 6. The energy from the hydrolyzed ATP molecules activates the hexose sugar and prevents it from being transported out of the cell. Next, we're going to look at the splitting of the hexose biphosphate, which is the end product of the reactions on the slide in front of you currently. Each molecule of hexose biphosphate is split into 2 3-carbon molecules, which are called triose phosphate, each with a phosphate group attached. Finally, we're going to look at the oxidation of triose phosphate to pyruvate. Although this process is anaerobic, it involves oxidation because it removes the hydrogen atoms from substrate molecules. Firstly, the dehydrogenase enzymes, aided by the coenzyme NAD, remove hydrogens from triose phosphate. The two molecules of NAD accept the hydrogen atoms, which are protons and electrons, and become reduced. At this stage of glycolysis, two molecules of NAD are reduced for every molecule of glucose undergoing this process. Also at this stage, 4 molecules of ATP are made for every 2 triose phosphate molecules undergoing oxidation. Both of the diagrams on the slide in front of you summarise the set of reactions. So here we can see the products of glycolysis. For each molecule of glucose, at the end of glycolysis, there are 2 molecules of ATP. 4 have been made, but 2 were used to kickstart the process, so the net gain is 2 molecules of ATP. 2 molecules of reduced NAD, and 2 molecules of pyruvate. So respiration of glucose has 4 stages. Firstly, glycolysis, which is what we've been focusing on in this video tutorial. Next, there's the link reaction, followed by the Krebs cycle and oxidative phosphorylation. The last 3 stages only take place under aerobic conditions. Under aerobic conditions, the pyruvate molecules from glycolysis are actively transported into the mitochondria for the link reaction. In the absence of oxygen, i.e. in anaerobic conditions, pyruvate is converted in the cytoplasm to lactate or ethanol. In the process, the reduced NAD molecules are reoxidized so that glycolysis can continue to run, generating 2 molecules of ATP for every glucose molecule metabolized.